is Ron Birchfield. This is my executive summary of research done in small molecule nanobody conjugate inducers of proximity. That's quite a mouthful. The executive summary is for Engineering 207 at Liberty University. Introduction and background. Brief explanation. Chemical-induced proximity, CIP, is a method that uses small molecules to bring two proteins together, dimerization. It enables control over cellular processes by creating temporal interactions. This approach allows for reversible and precise regulation of protein functions, thus controlling the proteomic interaction in the cell and the biofeedback loops that control many cellular processes. Limitations of traditional CIP. Traditional CIP approaches are transgenic and require modifications of an organism's DNA to introduce new traits, which can be time-consuming and complex. They also struggle with unligandable targets, proteins that lack natural binding sites for small molecules, and it limits the range and scope of the control that can be achieved. The rationale for integrating nanobodies with small molecules. Traditional CIP methods rely on genetic modification to tag proteins for interaction. This process is often laborious and time-consuming. Additionally, these methods cannot efficiently target unligandable proteins, which limits the range of proteins that can be controlled. These limitations restrict the versatility and the broader application of traditional CIPs. The concept overview. Definitions of small nanobody conjugate inducers of proximity. SNACP stands for Small Molecule Nanobody Conjugate Inducers of Proximity. In this context, proximity refers to the close spatial arrangement of two proteins. This closeness is chemically induced, allowing the protein to interact protein to protein with another body. By bringing proteins into proximity, their function can be modulated or activated. This technique is one that links a small molecule, a low molecular weight chemical, to a nanobody, a small chain of antibody fragments with high specificity to bring two proteins close together, inducing dimerization. This method enables rapid reversible control of protein interactions without needing genetic modification to the organism. Key concepts. Nanobody. Nanobody is a tiny antibody fragment derived from camelids, animals such as llamas and apex. It is a single dominant antibody consisting of only one variable region that binds specifically and strongly to the target proteins. Small molecule ligands. A small molecule ligand is a low molecular weight chemical that binds to a specific protein site. And cell-penetrating peptide. A cell-penetrating peptide is a short chain of amino acids that facilitates the entry of molecules into the cells. And medicine is no good unless you can get it where it needs to go. The general advantages of this approach. SNACP allows for quick and easy control of protein interactions. It works without changing the cell's DNA. It also affects proteins that don't naturally bind to small molecules. Additionally, it's very precise and flexible, and it means that only the intended proteins are affected. Its part can be mixed and matched, making it useful for many cell applications. General purpose of the chemically induced proximity factor CRGT. The CR10, it refers to the peptide that helps it get into the cell. SS represents a disulfide linker. The GBP is the binding nanobody, and quite often it has a fluorescent or radioactive protein to it so it can be tracked. And TMP stands for trimethylform, a small molecule ligand. Structural design. GFP binding nanobody, a small antibody fragment that specifically binds to green fluorescent protein, a protein that emits green light under UV illumination. It is derived from a camelid antibody and allows for precise targeting of GFP-tagged proteins. TMP ligand, a small molecule that binds with high affinity to specific proteins. And again, the CR10 is the ring-shaped amino acid that helps the protein get to the target. The process of heterodimerization begins bringing two different proteins from a functional pair. In this system, one protein is tagged with GFP, which glows under a certain light, and the other with the other protein from E. coli, an enzyme that binds to specific molecules. The conjugate-induced proximity molecule has two parts. One part is the GFP binding, which attaches to the other protein, and the other part is the ligand that attaches to the other protein. When the SNACP enters the cell, it binds to both proteins, bringing them into close proximity and allowing them to interact, which can control or modify their function. Key experimental findings. Rapid dimerization means that once this molecule is added, it brings two target proteins together quickly. About half of the maximum interaction is achieved within 7 minutes in vivo. The time needed to reach, This process is reversible, so by introducing a competitor which disrupts the binding, the protein can be separated again, allowing for dynamic on and off control of proteomic interactions in vivo. Applications demonstrated. Protein positioning. This technology can direct proteins to specific parts of the cell. Signal transduction. By bringing signaling proteins together, it can start or stop communication pathways inside the cell, which can also affect extracellular communication. And ferroptosis, which is a type of cell death like apoptosis, which is really nice when you're trying to get rid of cancer cells. Latent close chemical induced proximity, which is also a name for endogenous targets. Targeting microtubular nucleation factor. Targeting microtubule nucleation factor TPX2 means directing or activating this protein that plays a crucial role in the assembling of microtubules, which are essential for cell division. By targeting this, scientists can potentially control or interfere with cell division, which is important for cancer treatments. It allows effective shrinking of tumor cells. Oops. One moment, please. The design combines three parts: the TXP2 specific nanobody, which binds to the target protein, a cell cycle penetrating peptide that helps it get into the cell, and a CAAX box, a short amino acid sequence that directs the molecule to attach to the cell membrane. This signals for a modification that anchors the molecule. The mechanism works by anchoring the TXP2 specific nanobody complex to the plasma membrane using the CAAX box, which promotes membrane localization. This sequesters the TXP2 protein, a protein that normally helps initiate microtubule formation, away from its usual location, thereby inhibiting its function and reducing cell division. Experimental outcomes. Experimental outcomes show that sequestering this molecule leads to reduced cell proliferation in live animal models. This effect translates to tumor suppression, meaning that tumor growth was significantly inhibited. This is significant because it increases the efficacy, potentially, of traditional medications to target cancer cells in living organisms. Comparative advantages and therapeutic implications. Comparison with traditional dimerizers. Traditional dimerizers like rapamycin, ABA, and GA3AM also brings proteins together but have limitations. For instance, rapamycin often shows a hook effect where too high a concentration reduces its effectiveness and works within a narrow concentration window. ABA and GA3AM can face challenges with reversibility, which requires specific conditions to function optimally. In contrast, the SNACIP system offers rapid, reversible, and dose-dependent control of protein interactions without needing genetic modification, making it more versatile and easier to use to tune into different applications. The benefits: reversibility. They allow for protein interactions to be turned on and off easily. This is very good in things like, that's outside of this paper. Rapid kinetics, they work quickly. About half the maximum effect is reached in about 7 minutes. And versatility, their molecular design lets them be adapted to target different proteins without altering the cell's DNA. Potential novel cancer therapies and other applications. This protein and the process offers promise for novel cancer therapies by targeting proteins that traditional drugs cannot easily affect. They can sequester key proteins like TPX2, which are involved in cell division, and slow tumor growth down in living organisms. Additionally, this process can be adapted to regulate other cellular processes such as protein degradation or signaling transduction. Their rapid, reversible, and molecular nature makes them versatile tools for a range of therapeutic applications. Conclusions. Summary of major contributions. This paper presents the small molecule nanobody conjugate inducers of proximity as a new tool for controlling protein interactions without transgenic modification. Rapid and reversible control. Targeting heretofore untargetable proteins and modular and versatile design, which is very nice for controlling the plethora of intercellular proteomic interactions and implications for therapeutic applications. The rapid, reversible, and adaptable nature of this process holds significant promise for developing novel cancer therapies for broader applications in regulating cellular functions. Future potential. Targeted protein degradation. This could be adapted to tag specific proteins for degradation. This process of marking a protein for breaking down by the cell's recycling machinery, the lysosome. This would allow for the precise removal of harmful overactive proteins, which is significant. Spatial temporal control of signal. Their modular design also opens the door for controlling when and where signaling occurs within cells, which is very nice. Together, these advancements could lead to next-generation therapies that address diseases like cancer and neurodegeneration by managing protein levels and signaling pathways with high precision. Implications for engineering better medicines. This technology offers a new way to engineer better medicines. Oh, God bless America. It enables precise control over protein interactions without changing the cell's DNA. This method can target proteins that traditional drugs cannot easily affect, allowing for more specific therapies. Additional SNACIPs can regulate when and where signaling occurs inside cells. Spatial temporal control, which is crucial for fine-tuning cellular responses. In the future, these capabilities could lead to novel treatments with higher efficacy and fewer side effects, broadening the therapeutic strategies for complex diseases like cancer and neurodegenerative disorders. Thank you.